All right. Can you see my screen? And you're not
Yes.
seeing the video, people's faces in the upper right corner, are you?
No.
No.
Okay, there we go. There we go. All right. So, yeah, tonight I'm going to talk about some research I've been doing into the resilience of mosses. My original interest was how mosses tolerate desiccation and how they can recover so fast and et cetera. And then I read an article about these Nevada samples that had stayed in that herbaria for decades and they were able to resurrect them. So I just wanted to understand more about it. So I started doing a bunch of research, and then this talk is going to be broader than just the desiccation side of things. It's going to be just kind of the more broader topic of resilience of moss, but it includes the desiccation. So let me, let's see, go down a page here. For some reason, it's not letting me go. It's not liking my mouse trying to advance the slide. Maybe I can just click and it'll advance the slide. Okay.
Stacy, are you recording? I didn't see
Yeah.
the recording.
Yes, it says it's being recorded.
Okay.
Yeah. And we see your slide, Andy.
Okay, now the arrows are working. All right. So mosses have been around a long time, as y'all know. I did a little looking into this, and it's about 500 million years. So they've had lots of time to hone their survival skills, and they've been through, five of the Earth's mass extinctions, uh, wildly varying atmospheric content and temperatures, et cetera. Uh, and so this is just kind of the setting the stage for what they've had to survive. One particularly close call was this column about 300 million years ago, where we had the opposite problem of what we have today, where the CO2 actually dropped enormously down to possibly as low as 180 parts per million. Because moss rely on C3 photosynthesis, they don't like it below about 150 or so. So they're getting pretty close to, where they're going to have trouble breathing. Other plants like grasses would probably do okay. They use more, I think, I don't know if I'm getting the C3 and the C4 backwards, but the photosynthesis they use can work a little bit lower. But let's go ahead and move on here. So um, when I start getting into all these different mechanisms, I'm going to tap into a lot about how mosses propagate. And I started discovering that, oh my gosh, it's enormous, the number of ways that they propagate. I've got a much better drawing coming up. But first, I just want to say, if you just look at the two broad categories, the sexual reproduction, and we're always talking about the sporophyte and the capsules and things like that. And then we've got the vegetative reproduction. Um, but this is where things start to get wild. If you watch science fiction, you're familiar with the trope where the bad guy, like in Terminator 2, gets blown to bits and then reconstructs itself from the pieces. And well, Moss can not only do this, but they've got more ways to do this than all the science fiction movies I think I've ever seen. So I've got a diagram to kind of show some of this. This is for visualization only. No real moss looks anything like this. But what I've tried to show here is all the different ways that moss can reproduce. And so, there's obviously the capsules and the spores, and the spore germinates, and then this really thin little thing called the protonema comes out, and then buds form on that. And then those buds then grow into the full plant. But the mosses also have a bunch of other ways of reproducing, and one of them is you can break off a piece of just about any piece of a moss and stick it on the soil, and it can grow a new plant. And moss are so good at this that you can put them in a blender and spread that slurry over the soil to create a large patch of new moss. This works so well that it's a common technique that's used in both research and in restoration. So let me move on a bit here. Now, in the lower part of this picture, I've shown a leaf and I've shown a little protonema, or there's a couple of them, so we'll use the plural, protonemata coming out. And it turns out that it only takes a single living leaf cell to produce one of these. So you can see why the blender technique works so well. This ability for a single leaf cell to produce a new plant comes in handy if most of the plant has been killed by physical damage or drought or UV, freezing or heat. You don't even have to have a full leaf. You only need a couple of cells on that leaf if you want to, and you can create a new plant. So, and that's going to play into some things I'm going to talk about a little bit later. Okay. Now, I said almost any part of the plant can reproduce by fragmentation, but I ran into one reference in a paper that totally blew my mind. And this is the possibility of diploid moss parts producing the protonemata. So for a quick review, remember that moss spend most of their life in the haploid phase. You got the rhizoids, the stem, and the leaves, and these only have one copy of DNA. And if they split up and make more plants, those plants are clones. So they have the single copy of DNA as the parents. And as most of us know, the sporophyte parts, like the seta and the capsule, are diploid. They contain two sets of DNA. So how is it that part of them could be broken off and produce a haploid offspring vegetatively? I mean, they would have to undergo meiosis somehow. So it is extremely rare. And more commonly, when the diploid parts of the sporophyte do produce plants, the plants are sterile diploid plants. But it has been shown in the lab that it is possible for some species of moss diploid plants to actually undergo meiosis instead of mitosis to produce haploid protonema, which then will grow into new plants. So I'm still, my mind is still blown that that's even possible. It's like, how in the heck is it doing that? But anyway, so now let's go on to stuff that's a little bit less extreme and talk about something called propagules. So in addition to just breaking off parts of the moss, and those can create new plants, moss have three types of propagules that they can make. So what's a propagule? Well, it's a vegetative structure that is capable of detaching from the parent plant and creating a new plant. Moss have three types of propagules, and you won't, again, I'm showing all three on one plant here. I'm not sure if there's any plant that can have all three, but anyway, and what they have and where on the plant they occur varies by species. And so last month, I remember Stacy showed pictures of a moss that had gemma on the end of a stem. And in the picture here, I've shown them more along the leaf axis. And I've got a really good picture of this Oedipodium gryphatheum. It's from Wes Callahan. And this shows what the gemma look like on a leaf. And so these little tiny things, they can just get knocked off even with raindrops, and then they'll fall on the ground, and then they can create new plants. On some moss, they grow in special receptacles called gemma cups, and I think these gemma cups are pretty common on liverworts. But anyway, so let's move on here and get back to this drawing. Another type of this propagule are bulbils, and they're more robust than the gemma, and they even can exhibit, if you look up close, the beginnings of leaf structures on them. They're a little bit harder to break off, but they're a little bit further along in terms of becoming a new plant when they do break off. Let's see, the third propagule is a tuber. They're less common than gemma and bulbils, and they usually form below ground. So they aren't spread, away from the plant by wind or rain. And they have two ways of really helping survival. When they're attached to the plant, they can actually store nutrients and liquid that'll help the plant recover after drought or freezing. But they can also sprout protonemata and grow into an entire new plant. And in fact, I'll give a really good example of an extreme version of that a little bit later when I go through the case studies of of mosses doing some pretty wild things. Last month, I was curious about the different structures that I was seeing at the end of the capsule, and a bunch of you helped me understand what's going on. I thought I was seeing three things, but I'm actually seeing four things. And so these are some pictures of my moss. That picture at the very beginning of the presentation, that's my little moss garden I've got growing in a small kitchen dish in our plant room. And these are some moss that are putting out sporophytes right now. So, you've got the calyptra, and that is kind of fell off, and it's still a little bit stuck to the capsule there in the one in the back. Then you've got the operculum, so that's two. And then the other two things I was seeing are these outer and inner peristome teeth. And I was actually able to watch these open and close. I could put water on this thing, and those outer peristome creep uncurl and then cover up the inner peristome. Let me go to a slightly exploded view here so you can
Oh,
see what's
wow.
going on.
So
And
cool.
so what's going on is that these inner peristome teeth, they don't really change as the moss gets wet and dry and wet and dry. They just kind of, at least on this one, as I was watching it, they pretty much stay the way they are. And they help hold the spores in so the spores don't all come out at once, because you kind of want a timed release, because sometimes it's windy and sometimes the wind's coming from one direction, or you want those things to spread far and wide. So you want them to come out, you know, paced over time. But you also don't want them coming out when it's wet, because you want them coming out when it's dry, and they can blow in the wind. If it's wet, the water will just kind of knock them below the existing moss. So that's the function that these outer peristem teeth perform. And I said, well, I've got to get a picture of these things releasing some spores, and I did. And hopefully you can see to the left, there's a little bit of a wind current in the room. Those are spores dropping out of this sporophyte.
That's such a cool photo. Oh my
Yeah.
God. Was
So
it windy or
cool.
wet, or how did you capture that?
So what I did, let me tell you the story behind this. So I was looking at these things, wanting to get some pictures, and I had a loop out, just a 10x loop, and I was kind of looking for a good one to take a picture of. And while I was watching, I saw this puff of spores come out, and I'm going, oh my gosh, I've got to get a picture of that. And so then I got my camera set up and everything, and I thought, well, maybe I wasn't seeing any coming out. I thought maybe they're more likely to do it right after they dry out instead of before or something. So I got some wet, and I waited an hour, and then as these outer peristome teeth curled completely back, I was watching under the loop, and I saw some spores drop. I said, okay, great. So then I had a camera set up, and I sat there, and for about 15 minutes was taking about 2 pictures per second. And I didn't have the time to go, there's settings on the camera that can do this automatically, but I just sat there and pushed the shutter and click, 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 click. And when I got done, I went back and looked through all my pictures. And it seems like about every 15 seconds, I would catch a few spores coming out. And this was the best picture of the spores.
That's
So
fabulous.
So now I'm going to change gears a little bit and go through a bunch of case studies of really interesting moss that live in extremes or do extreme things to survive. Okay, this one's called Takia, and it's the oldest moss genus, and it's been around. From molecular studies, they think it's been around for about 390 million years. The oldest fossils are about 165 million years back, but that's still pretty darn good that we still find it today. The leaves are a little different than traditional moss leaves. The top and bottom, there's no kind of distinction the way these leaves are. And it's found in an interesting place. One of the places it's found is on the Tibetan Plateau at 13,000 feet. This is the site of the fastest warming recorded on Earth right now, 0.77 degrees per year from 2010 to 2021. I mean, that's pretty darn scary if you think about it. I mean, I was reading through this stuff, and it was like, holy cow, that's like 8 degrees a decade or something. I mean, so anyway, but these things have this really incredible ability to tolerate ultraviolet light. And there's some experiments that have been done. In one experiment, they blasted it with 10x the max UVB in particular that occurs where it lives. And this Takia was unaffected. And they were running some other moss by, and they died in 24 to 72 hours. So you say, wow, how can this thing tolerate that much ultraviolet light? And well, it's got three strategies. And so the first thing, you got to stop the UV from getting in, because once it gets in, it does two bad things. It can directly damage the DNA, and it creates these reactive oxygen molecules that can also damage the DNA, but it can also disrupt other cellular functions. And we've all heard about foods containing antioxidants, that are supposed to be good for us. And that is what the moss is doing in step two. So there's always going to be, and so what in step two we've got is all these enzymes that are out there gobbling up or, taking out the reactive oxygen species before they can do damage. So there's always going to be some damage, though, and that's where the third strategy comes in, which is the repair mechanisms. And Takia has some strong things. In fact, it has some DNA repair mechanisms that are more common in higher plants and animals. So to put some numbers to this, here in Fort Collins, we get about 20 to 25% more UVB than you would get at sea level. At 13,000 feet where Takia lives, it's getting about 40 to 45% more UVB than at sea level. But there's another reason that Takia probably developed this strong UVB protection. And that's because it was around for several of Earth's ozone depleting mass extinction events. So remember from that first slide where I talked about the 5 extinction events, and they're caused by different things, but both volcanic activity and meteor impacts can disrupt the ozone layer, and that allows more UV through until it can be regenerated. In the case of the Chicxulub impact 66 million years ago, that's the one that nuked the dinosaurs, UVB radiation probably rose 2x to 5x what it is today on Earth. So, and of course, Takia just survived right through all that. So it probably, it honed its techniques while this was all going on. So you might be wondering about desert mosses, even in New Mexico and Colorado, like Centrichia ruralis. And as it turns out, yes, Centrichia ruralis also has strong UV protection, starting with a waxy coating on the leaves and the stem that help block UV. And it also uses enzymes to neutralize reactive oxygen species. It's not quite as tolerant as Takia, but it's a lot more tolerant than mosses that are more commonly found in forested, wetter areas. So now that we've talked a little bit about intense light and intense ultraviolet light, let's consider a moss that can live on the least amount of light, okay? This is Justica Panata, also called Goblin's Gold. It can live with the lowest light of any moss, 0.02% of full sun. Okay, so if you're a photographer, that'd be 6.5 to 20 lux. This is like 10 minutes after sunset. Okay, this plant can survive. So what happens is when the light is low, let's just start with the spores. Let's say the spores get dispersed and they go partially into the entrance of let's say a cave like this. The spores start producing the protonemata, okay, and those things start spreading in search of light. And they don't produce buds or plants if it's dark. But what they do produce is this other cell that can harvest light really efficiently. And I've drawn a diagram of what it looks like right here. I drew this diagram from some microscope pictures I found online of what it looks like, but I wanted to better illustrate how it works. The top of this cell acts like a lens, and so the light is focused then on the back of the cell, and the chloroplasts actually move around in the cell to that spot. So if the light moves across the lens over a period of 3 to 4 hours, the chloroplast will actually drift so that they're in the focal point. In other words, they're trying to get the most amount of light they can harvest like, for example, as the sun is moving across the sky or they get disrupted or whatever, okay? And then once the protonemata have grown far enough to where they can find more light or better conditions, they will start to produce buds and produce the plants. And in the upper right here is what the gametophyte looks like, okay? And these aren't that rare, actually. They're found across the northern hemisphere. And they actually don't compete well with other moss that are more traditional, that can tap into the normal amount of light. The Japanese are really into this particular moss. There's even a monument to this in Japan. Okay. Um, so next we're going to start talking a little bit about desiccation in water and stuff. Okay. So, we've got no vascular system, no roots, and no stomata. Well, no traditional stomata on the body of the plant. Of course, you can get these stomata things on the capsules and stuff, but in the main plant, how do you move water and all these other things? All right. So, and this lack of a vascular system is also why mosses don't grow very tall, because they can't pump nutrients and water up and down their stems. The early vascular plants on Earth grew to enormous heights, the equivalent of a plant dinosaur, if you will, which is where the Earth's coal deposits came from. But because the moss doesn't have that vascular system, they have to stay small. How many of you know what the tallest freestanding moss today is? Does anybody know?
Dawsonia.
Yes, Dawsonia. Very good.
These dorks, we don't stand a chance with you.
I love it. Well, I hope I'm telling at least a few of you some things you don't know. Yeah, it's in New Zealand and Australia, and it can grow to 2 feet tall.
Yep.
Okay. But moss still have effective ways of moving water and gases up and down and across the plant. They can use diffusion and stuff like that. In this picture, what I've got a picture of is the awns. Those awns, they don't have chloroplast. You go, well, why does the moss bother to produce this big long thing with no light gathering? Well, that's because it's collecting water. And then it has little ridges and things that help collect, dew or fog and then direct that water down on into the leaf. So now let's start getting into some desiccation stuff here and desiccation tolerance. So mosses are well known for their ability to dry out and come back to life. And this is found in most bryophytes. And it isn't unique to bryophytes. Some vascular plants and even animals like the African bullfrog have ways of shutting down for years when water is scarce. Of the vascular plants, ferns are probably the most desiccation tolerant. And most flowering plants, they don't have any desiccation tolerance. Interestingly, in vascular plants, it is the vascular tissue itself that crashes the hardest when they start drying out. So I guess their feature is their downfall when things get dry. In moss, on the other hand, their internal moisture, they tend to track the external moisture. They're not storing up a whole bunch in tissue. And this tracking is a property that's called poikilohydry. Okay. So when desiccated, the moss shuts down completely. I mean, there is no measurable metabolic activity, no photosynthesis, no respiration, and it can be shut down in extreme cases for decades and resume living after rehydration. That African bullfrog I mentioned a minute ago, it does not completely shut down. Even though it can stay dried out for a year or two, it still has a heartbeat of 2 beats per minute. So can you imagine a heartbeat every 30 seconds? Vascular plants similarly don't completely shut down, but they store water and continue metabolism in some parts of the plant at a greatly reduced rate. So how do mosses do this? Okay, well, the cell membranes have a higher amount of fatty acids, and and when they dry slowly, they're more likely to remain viable. Because if a cell membrane leaks, the cell's going to die. inside the cell, there's another thing that happens, and the sugar content goes up. And that helps hold things in place as well, so that you don't get this, it'll still shrink, but it's not shrinking so much that things are breaking. And while moss can shut down in dry times, they can't afford to spend too much time shut down, or they're going to miss the opportunity to grow and reproduce. So especially in desert areas like where we live, when you get a rain, you got to come back quick. And I'm going to talk about that next, but I want to make sure I've covered the points here on the slide. There's, I guess there's one other thing I wanted to mention here is that there's two types of desiccation tolerance. There's one called inducible, and that means it's mosses that they can tolerate it, but they got to have a little bit of warning. They've got to either have some cycling before they really get hit hard, or you have to dry them fairly slowly. And then there's other moss, like the ones that are more common in the desert, that they don't really require any training at all. You dry them up, no acclimatization, and they're fine. These constitutive ones, the ones on the right here, they tend to be slower growing, but they also tend to rehydrate quicker. And we might ask, how quick? So this is a chart about how quickly things boot back up after the moss gets wet. The first thing you need, of course, is you need to get some energy going. And the photosynthesis can start in a matter of seconds in some mosses after you add water. Now, once the photosynthesis is going and you're producing ions and things or whatever, you got to start making energy that the rest of the plant can use. So ATP synthesis starts fairly shortly after photosynthesis. Next thing, minutes to hours, ion transport and membrane repair. When it dries out and gets rehydrated, there's always going to be some damage. So you got to start putting things back together again. And that's followed with the protein synthesis. And then usually in a day to days, you start having cell growth and cell division again. All right. Now, this varies a lot with species and exactly how slow it was dried and things like that. Okay. So now I'm going to talk about the record is Centrichia ruralis for desiccation and then bringing it back to life. And there's a beautiful picture online, but I don't have permission to use it. And everything in this slide is not copyrighted. So this slide, we can put this deck online. We can share it with people. And all my drawings, I let anybody use them and pictures, use them for anything they want. So in 2016, let's get back to this. This Centrichia ruralis was taken out of storage at the University of Nevada, where it was kept at around 70 degrees, around 32% relative humidity for 20 years and 3 months. And they added some water to it. They took 58 shoots and they added some water. They put them on a sterile medium and they added some water. And it wasn't very quick. Remember, most of those cells are probably dead. But remember, it only takes one cell from one leaf to produce a proto-anema. But surprisingly, of 58 adult shoots, 38 were able to regenerate. And there were an average of 3 regeneration points per shoot, with the average first generation occurring on day 17. So this is certainly not as quick as moss come back if we're out on a hike and pour a little water and we watch it, over a minute or two turn green. These things recovered very, very slowly, but they did recover. And the leaves were also able to produce new protonemata. Here's an interesting statistic, though. This actually surpasses the record for desiccated bryophyte spores, which is 16 years. And I would have thought that spores, you think of seeds can last, some seeds at least can last a long time. But this record for the Centrichia ruralis exceeded desiccated bryophyte spores. So another thing that probably helped these was the fact they were stored in the dark. Not only does UV light damage, even visible light can slowly damage plants, if they're sitting there just inert or whatever. So I came across, I got to tell you one other thing before I get into this paper. At the end of the presentation, I've got 3 pages of references for all the papers I read to prepare this talk. I started reading this one that was just fascinating. It had all this data. And when I got to the end, I realized this paper was written in 1898. So there was good science going on in mosses back in the 1800s, and it was relevant for this talk, and it's in my references at the back. But anyway, I came across this. This was one of the other unusual. These people decided, this Chinese team decided, well, let's just beat the heck out of a moss and see what it can tolerate. So these aren't natural conditions found anywhere on Earth. This is just them testing it out. So the first thing they did was they hit it with gamma rays, and this is pretty amazing. So the lethal dose, gamma rays are measured in a unit called a gray, and the lethal dose for humans is 4 grays. They hit this Centrichia canenervis. Oh, I don't know how to pronounce it, but they hit this thing with 4,000 grays, and it survived. And that's what these pictures are right here. Day 15, you can see it looks pretty much dead, and by day 60, oh my gosh, it's putting out a bud. To compare this, everybody hears about tardigrades, and how you can put them in space and hit them with radiation. This is a similar amount of gamma rays that a tardigrade can tolerate. I wonder if it's no coincidence that tardigrades like living in mosses. Anyway, they also did a bunch of other things. They stored it at minus 80 C for 5 years, and it was able to recover. They dipped it in liquid nitrogen for 30 days and pulled some moss out, and they were able to reproduce. And then the last thing they did, they said, well, what about Mars? What if we simulate Martian surface conditions? We're going to simulate the low pressure. Mars has almost no atmosphere. It's almost a vacuum. They simulated the gases, the pressure, the UV, and even the wild temperature swings that the surface of Mars get. They did this for 7 days, and then they put the moss back in some sterile sand and, put some water on it. Oh, this was done dry, by the way, the Martian conditions. And then it actually came back to life. So
Crazy.
yeah, it's unbelievable. So now we're going to get back on normal things happening on Earth. And this is a moss that was frozen for 1,500 years. So this is on Cygnia Island. I've got a picture of Antarctica up here, and it's a little island off the tip there. And in 2014, this team from the British Antarctic Survey took a sample, and they took a core. So this is just like a round core that goes way down into the ice where there's living moss on top, all the way to the bottom. Now there's continuous moss tissue throughout this whole 54 inches. And so you say, how do I know that this moss on top wasn't sending some nutrients and stuff down to the bottom over these 1,500 years to kind of keep it going and stuff like that? So what they did was they took this and they carbon dated the layer above the layer that they're going to run the tests on. And when they carbon dated that layer, it came up with 1533 to 1697 years before present. Okay. And this is after calibration. Yeah. If you want to know something about, I probably spent 3 hours just learning the intricacies of how radiocarbon dating works, which is fascinating of itself. But anyway, they took the plants, and after 22 days, some green protonemata started coming out of the brown rhizoids. And so they said, wow, protonemata. Now we also got to make, they look at it under a microscope and they don't see any spores, but say, we got to make sure that, there's not some other explanation for this. Well, it turns out that sporophytes of this Corisodontium acephalum are not found in the maritime Antarctica. So they feel like it's pretty unlikely that there was some hidden, preserved spores down there or something. But even then, 1,500 years, that's pretty incredible. Okay. So that's what I've got for you for tonight. In summary, we've talked a lot about the reproduction and all the different ways moss reproduce. Talked about high light, low light, drying out, freezing, and stress is not found on Earth. And so there you have it. That's what moss have been up to the last 500 million years. figuring out how to persist on our planet. And these are my 3 pages of references right here. I found that if you create a free login on this jstar.org, you can review 100 free articles a month. I was trying to figure out how can I not spend a fortune getting to a whole bunch of these articles behind firewalls. Also, I joined the British Bryophyte Society. It's only 25 pounds per year. I think it was a little closer to $30. But you can view all back issues of the Journal of Bryology. And I found a lot of interesting information back there as well. And there you have it.
That's
That
Wow.
was fantastic.
Oh,
That
wow.
was amazing.
Thank you so much.
I learned so much.
Good.